In this video, I will be discussing the PD-L1 protein and its impact on the rising cancer rates. Over the last several decades, cancer has become a predominant disease all across the world and is currently the second largest cause of death. Unfortunately, cancer's increasing rates don't appear to be slowing down anytime soon. This is causing a huge concern for individuals everywhere. As you can see here, this is a graph showing the rates of skin cancer or melanoma cancer and how it has been gradually increasing over the last 2 decades. This increase in cancer could be due to multiple different factors, but a factor that we are aware of that is causing this new increase in cancer is the cancer cell's new ability to express PD-L1 to avoid cell death. When a cell becomes injured or infected, your body uses T cells to mark the unwanted cell with a protein called PD-1. PD-1 serves as a flag to your immune system that this cell must be killed in order to maintain homeostasis. This flag also signals to the surrounding cells to stop producing PD-L1, which is PD-1's ligand. But cancer cells have evolved to avoid being killed. Instead of shutting down their production of PD-L1, they begin to express an excess amount, which will then bind to PD-1, creating inhibition of the T cell. In this figure, you can see the gray section is the PD-1 protein, or the flag. The pink section is the PD-L1 protein, the protein that cancer expresses. And the purple section is where PD-L1 and PD-1 connect. You can see how tightly these molecules are bound together, with no space in between. The tightly bound nature of these proteins together proves why these tight interactions are a strong inhibitor for the cancer cell. This next image shows PD-L1, again in pink, and PD-1, shown in gray. These proteins are now shown in a skeletal form and separated rather than bound together. From here, we are able to focus on the attachment portion of PD-L1, shown in the dark purple. You can see this attachment portion extends outwards away from the protein in order to bind with the PD-1. Once these proteins are bound, the T cell will be inhibited, allowing the cancer cell to continue to grow. In order to simplify this information, we have broken down the biggest aspects into 6 simple steps. The first thing that has to happen is a T cell will travel through an individual's body looking for dead or infected cells. If a dead or infected cell is found, the second step will occur. This T cell will then place a flag, as we know as PD-1, on top of the cell in order to signal stopping production of PD-L1. Under normal conditions, this cell would be destroyed. However, as we know, this is not the case with some cancer cells. The fourth step that will happen is the cancer cell will begin to express an excess amount of PD-L1. Following the increased expression of PD-L1, step 5 will occur, where PD-L1 is able to inhibit the body's immune system response, allowing the cancer cell to grow. Finally, the 6th step in the process will occur. The cancer will then be able to freely spread and grow in an individual's body with no way of stopping this.
This is the EGF protein.
As cells die within our bodies, it is important that they are replaced, and it is also important that those replacement cells grow properly. EGF, or epidermal growth factor, plays a large role in when these cells are told to grow and divide. It is the signal that tells cells when to grow by binding to an extracellular receptor. Once EGF is bound to its receptor, a receptor nearby forms what is called a dimer. This dimerization is then formed through the transmembrane domain and into an intracellular domain. Once dimerization has occurred in the cell, a cascade of events occurs so that the cell may begin its growth. There are 2 forms of the EGF and EGF receptor complex. One is the inactive form and one is the active form. The The inactive form is when the EGF and EGF receptor are interacting, but the signal is not yet sent through the cell, and dimerization has not yet occurred. This can be seen as the EGF being purple and the receptor being green. The active form is when the EGF and EGF receptor are bound to one another and dimerization occurs. This can be seen here with two EGF signals being on the outside of the two receptors. EGF signals are colored red and the EGF receptors are colored blue. The problem that arises is when the EGF and its receptor are bound for too long. This causes the cell to grow uncontrollably and divide nonstop. This uncontrollable growth can be seen in the form of overlapping cells, cell bunching, and cells clumping together. Ultimately, tumors and other cancerous growths are made and could have detrimental consequences on the health of the affected person. Through our research, however, we are able to better understand the EGF and EGF receptor complex. This understanding will allow us to find solutions to stop this uncontrollable growth and ultimately save the lives of many.
In a healthy living cell, the mitochondria is the battery of the cell. You can see cytochrome C, which is in orange, in the inner membrane of the mitochondria. When the mitochondria is taking on damage and the membrane of the mitochondria gets damaged, this then allows the cytochrome C to escape. When the cytochrome C escapes, it signals that something is wrong with the cell and that the cell needs to die. The cytochrome C then binds to a molecule called APAF1. APAF, which stands for apoptosis protease activating factor, is shown in blue. The term apoptosis means programmed cell death, and the term protease stands for chopping up proteins. The cytochrome C is bound to the propeller-like structure of APAF1. Now these units will assemble together to form a bigger structure named the apoptosome, which is composed of 14 subunits, 7 of which being APAF1 and the other 7 being cytochrome C. Once this bigger structure is assembled, it can start the programmed cell death. When the apoptosome forms, it starts the degradation of the cell, where in cancer cells, this action of forming the structure is prevented and therefore prevents the death of the damaged cell. In cancerous cells, the degradation or destruction of the cell also does not occur, which means the cell will not end up being able to start its programmed cell death, thus allowing the damaged cell to continue on without being destroyed. This in turn gives the cancerous cell the ability to mutate and multiply, allowing tumors to form.
Telomeres are caps at the ends of chromosomes that protect the DNA from damage, which are made of repeating non-coding DNA sequences. As cells divide, the telomeres shorten after every division, but once the telomeres reach a critically short length, it signals for cell death. Telomerase is an enzyme that is composed of protein, shown in orange, and RNA, shown in purple, in which it elongates the ends of chromosomal DNA, shown in green, which are known as telomeres. To help prevent this shortening from occurring, telomerase is used to elongate the telomeres by using an RNA template and a specialized reverse transcriptase known as TERT. Reverse transcriptase is an enzyme that transcribes or copies single-strand RNA to DNA. The reverse transcriptase can be seen here in orange, and the RNA template shown in purple is partially hidden by the green DNA that is being elongated. This video shows the elongation process occurring, such that telomerase is adding the repeating sequence to the end of the telomere. In normal cells, the telomeres get to this critical length, ultimately resulting in the cell's death.
However, in cancer cells, there are mutations present that lead to high levels of telomerase. This abundance of telomerase allows for the telomeres in rapidly dividing cancer cells to stay long enough such that there is no cell death signaling and the cancer cell stays alive.
When cells in the body get damaged, the normal cell growth gets blocked, increasing cell division. Cancer is a cell disease that causes cells to divide uncontrollably and produce more cancer cells to the site of the damage. This increases the size of the cancer tumor in the body. Research has found that the p53 tumor suppressor helps protect the cell from this cancerous damage. This video focuses on the 1-TUP protein in p53 tumor suppressor and how it protects the cells. The overall structure of the p53 tumor suppressor contains DNA shown in orange and 3 molecules shown in aqua. These molecules are also known as the protein, and they are aligned in an asymmetrical unit. 2 of the molecules at the top and at the bottom of the protein have been removed from view because even though they bind, they weakly interact with the DNA helix. On the other hand, the middle domain molecule is very important while it directly interacts with the DNA. The p53 tumor suppressor contains a zinc atom, shown in pink, which plays an important role in bringing the DNA and protein together. This zinc allows for all molecules to have a tight relationship with each other and is what brings all the protein together. 4 important structures within the protein, also known as amino acids, interact with the zinc. These amino acids, shown in blue, consist of 3 cysteines and 1 histidine. Without the zinc atom, these amino acids would fold within the protein itself. The middle core domain interacts with the minor groove of DNA. This molecule can do this through an arginine amino acid in the protein. Arginine reaches into the minor groove and interacts directly with the DNA. The minor groove interacts with DNA, and arginine plays a critical role in DNA binding. Arginine is the most frequently mutated residue of p53 in human cancer cells. All in all, the p53 tumor suppressor comes into play when mutations are present in the cell. The goal of p53 is to prevent cancer from spreading. It does this by binding protein to DNA and halting the replication of cells through various structures.
Estrogen is a hormone that allows our bodies to grow in major spurts throughout our lives. It is a small, carbon-rich molecule that is built from cholesterol and most found in women. In women, estrogens are responsible for sexual development. They assist in the development of a single cell to a full-grown baby and also during puberty when children mature. Not only does estrogen encourage growth and development, but it also protects the heart and liver from cholesterol, strengthens the bones, assists the brain with memory and function, and has an anti-aging effect on the skin. Estrogen, which is colored green, differs from regular hormones because it passes directly into the cell and binds to its estrogen receptor, which is colored purple. When estrogen binds to the estrogen receptors, they pair up and form a dimer, which is shown here. Estrogen receptors must come together in order for growth to occur. The estrogen receptor is located in the nucleus of the cell, right where this lime green DNA is active. The dimer formation allows the estrogen receptor to bind to specific sites in the DNA. These sites are placed next to genes that need to be activated to stimulate cell growth. In certain forms of cancer, excess amounts of estrogen are released. This causes unnatural growth. One way to stop this growth is the use of a drug called tamoxifen. Tamoxifen is a small drug that resembles the shape of estrogen. Tamoxifen binds tightly to the estrogen receptor, blocking estrogen from binding because it is slightly bigger. When estrogen is unable to bind, the dimer does not form. Therefore, there is no signal for the cancerous cell to continue to grow.